Okay, so we're going to talk about the immune response. First of all, just talk about a couple of background things here. So what I've drawn here is we've got a cell, and on the surface of every cell, you have these molecules called a major histocompatibility complex type 1. The only kind of cell that doesn't have this are red blood cells. Every other cell in the body does. They were called this, first of all, histocompatibility because the molecule is a bit different in most individuals, and this is the reason why you can't get a kidney from anyone except a close relative, because the MHC molecules on somebody else are a bit different and you reject it. That is a bit of a misnomer because that's not what its role is at all. We'll see what the MHC's role is here. Right, so we've got a cell, and in a cell you have loads of proteins, and those proteins have a limited lifespan. And when they're finished, they get broken down in, right, so here I've got, I'm drawing a vesicle, and there's a protein in there. It will get digested and broken into little bits, okay? It's a different color. It's broken into little bits. Okay, now this black protein here, this is the MHC protein. So that will, first of all, it will be inside the cell. And I'll draw that a little bit bigger. So it'll be in the vesicle inside the cell. And it's kind of like, it's got a cleft in it, and there's room there for a little polypeptide to go in there. Okay, so any polypeptide, not specific for any particular one, little bits of polypeptide will go in there in the cleft. And then once you've got a bit of polypeptide in the surface, this vesicle then migrates to the cell surface, to the cell membrane. And so what you end up with lots of these MHC class 1 molecules with little bits of peptides stuck in it. And those little bits of peptide are called antigens. And this whole process is called antigen presentation. So in a normal cell, you will have lots of these MHC type 1 molecules on the surface, each one with a little peptide in there, and nothing will happen to that cell because that's bits of you, bits of self, and so it will be recognized as being okay and nothing will happen. However, say if you get a viral infection in one of these cells, we'll talk about, so you'll get a virus here, and you get a little bit of virus. I'll do it in a different color, the virus one. Okay, so you get a little bit of virus protein getting broken down. That will end up getting presented there. Now, that will be recognized as being not quite right. It's a sign that the cell is infected by a virus and your immune system is going to come along and kill that cell. Okay, so that's antigen presentation. It happens all the time, and most of the antigens that are expressed are recognized as being yourself, and you don't do anything to it. Okay, now we just need to talk about a little bit about different kinds of white blood cells which are involved in immunity, and you've come across some of them before. So you will have come across phagocytic cells, like macrophages. The ones we're going to talk about today are lymphocytes. If you look at a lymphocyte in the microscope on a blood film, you see it's got a very big nucleus which almost fills up the whole cell and not much cytoplasm. Now there's two kinds of lymphocytes. They both look the same. You have B cells, B lymphocytes, and they're involved in making antibodies, B cells, and deal with them in a later video. And we have these ones, T cells, and they're more involved in dealing with viral infections and things like that. Okay, moreover, you can split the T cells into two different types. A certain type, they've got a molecule on the surface called CD8. So it's got a little protein on the surface called CD8, which is incidentally the protein that coincidentally the HIV virus binds to. And if you've got CD8, it means that they're kind of cell called a cytotoxic Sorry, cytotoxic T cell. Cytotoxic means they kill cells. Usually they kill cells that are infected by viruses, your own cells. And there's another kind, and they don't have a molecule CD8, similar molecule called CD4 on their surface. And they're called T helper cells. And we'll talk about those in a later video. We're going to talk about cytotoxic T cells today. Okay, so let's see what happens if you get infected by a virus then. So here's a cell. Normally, a virus is it will attach to a receptor on the surface of the cell, trick its way in. So the coronavirus binds to a protein called ACE, angiotensin converting enzyme, which has nothing to do with the coronavirus, but it tricks its way in, gets into the cell. Once it's in the cell, it then subverts the machinery of the cell, and it makes the cell stop doing its own job of making its own proteins, makes it make a whole load of viruses, and then eventually the cell gets full up with viruses, it bursts open, and you get loads of new viruses come out, and they go infect other cells. But before that happens, if the virus gets broken down in the cell, then a little bit of that virus will get presented in the Mhc molecules, okay, and they will be recognized by one of these CD8 cells. They'll recognize it as being shouldn't be there, and that will initiate killing the cell. Right, now how does the CD8 cell do this? Well, it has something on the surface of it called a T cell receptor. Right, a T cell receptor is a protein, sometimes called TCR, and it can recognize, there are millions of different T cell receptors, okay, all different. They can all recognize slightly different antigens. Well, how can that come about? Because there's only one gene for the T cell receptor, so there should only be one protein. Well, let's talk about how that happens here, which is right. So what I've got here, this is a T cell, a progenitor cell. In an embryo, okay? Okay, so the developing embryo has got a T progenitor cell. Then what happens is you get a rearrangement of the DNA. So the DNA is spliced. So basically the DNA, which codes for the T cell receptor, gets chopped up into little bits and stuck back in slightly different ways. And that gives rise to, and every single one of these is different. So it gives rise to all of these different cells, and they're all similar, but they've all got a different T cell receptor. Okay, so different T cell receptors, and there's literally millions of these. So you've got millions of different T cells, each one with a different T cell receptor. It will be a little clone of cells, actually, it won't be just one, because that will then divide and make its own little batch of, 100 or whatever, its own version of itself. They've got different T cell receptors. Now, in the embryo, what happens is any of those the ones that recognize self-antigens, okay, so that's the ones which I've drawn up here in this first diagram I did. So all of these things, these are all self-antigens. Yeah? The only one that's not self-antigen is that one there, which was, you said, came from a virus. So anything which is a self-antigen, and there'll be lots and lots and lots of different of those, tens and hundreds of thousands of them. If it recognizes self-antigens, what happens is it gets wiped out by a process of cell. It's called apoptosis, which is the cell is told to kill itself. Okay, so these ones here are the ones that recognize self, and so they get wiped out, and they don't last into the mature animal. Okay, all that's left here are these ones, all of these T cells, and they recognize non-self. and there's literally millions of different versions of those, and they could recognize almost any molecule that could ever be made. So for example, the coronavirus, it only came about in 2019, but obviously people have had clones of T cells in their blood, in their bone marrow, which were just waiting there, ready to recognize something that shape and ready to spring into action. Well, the coronavirus may not have happened, or you may not have got it. You've still got that cell there, and you'll have millions of other kinds of T cell clones which will never do anything. They'll never recognize anything, but you've got so many of them. They're there. They can recognize any new molecules which come along. Okay, so let's see what happens then. So we have now got our cell here, which has been infected with a virus. Okay, got a cell which is infected with a virus, and the particular T cell clone that recognizes this antigen is going to come along. So that's a very specific antigen which is presented. Virus antigen, which is okay, and the T cell comes along, and here, so I'll draw the T cell, draw it in blue. This is our cytotoxic T cell. So it's got CD8 on the surface of it. And here's its T cell receptor. It's obviously not really very to scale. And that's going to recognize that whole major histocompatibility group with the foreign viral antigen in there. So that binds to the T cell receptor. And then what that will do is, so it binds to the Mhc complex, and that activates the T cell. It activates the cytotoxic activity. Okay, so what the T cell will do, first of all, it will produce molecules called perforins. They perforate it, as the sound sounds. They make lots of little holes in the cell membrane of the target cell. So that's the infected viral cell. You've got lots of little holes in it, and that causes the infected cell to die. Now, this infected cell dies, it can't make any virus, so you don't get like millions of new viruses maybe which can go and infect another one. So it has to sacrifice one cell to stop it being taken over into the all virus factory. Now, this can happen with just the cytotoxic T cell itself, but what makes it better is if you have another kind of T cell, a T helper cell. And we'll talk about them later, but a T helper cell, what it does, it makes a little molecule called interleukin 2. Sometimes it's called IL-2. And that acts as a sort of hormone which only acts on it's a protein. It only acts on a very, very local level though. So it doesn't get into this blood circulation and go right around your body. It just acts at a local level. It's produced by T helper cells in the locality. And what it will do, it will stimulate the cytotoxic T cell. So that's the CD8 cell. And it will stimulate it to become more cytotoxic, and it will also make it divide. So you'll end up with a clone of those little cytotoxic T cells that can actually recognize this viral antigen. So they're perfectly made just to deal with that viral antigen. So you get a whole load of them made, and they'll go all over the body and attack any cells which are infected with the virus. Okay, so that is how sometimes it's called cell-mediated immunity. It doesn't involve antibodies. You won't have come across this sort of immunity before at GCSE. It doesn't get a mention, but that is how viruses are dealt with by the immune system.